Again to our weekly agriculture and biotech webinar. As you know, this is organized by Food for Sustainability Academy together with our Grow partners. For those that don't know me, I'm Paula Costa. I'm the director of Food for Sustainability Academy, and today we're going to take a closer look to how living soil and water communities, and we'll go from aquatic fungi to bioinoculants and soil transplants, how these living soil and water communities can help us bring our soils back to life. And our webinars, they build this connection between advanced microbiology with very practical questions. And the questions for today is how do we restore function, resilience, and productivity in degraded systems? And what does this mean for day-to-day decisions in the fields? And to help us answer these intriguing questions, we are joined by a fantastic group of speakers, which I'm very pleased to introduce. So we have Paula Castro. Paula is a professor at Católica University of Porto, and Paula will be exploring how plant microbe solutions and bioinoculants can reestablish this healthy soil and these living networks. Then we'll be followed by Anita Perkins. She's a mycological biogeochemist at the Aquatic, Botanic and Microbial Ecology Research Group. She comes from the Balaton Research Institute, and Anita will open a window into the world of aquatic fungi and their role in the ecosystem functioning. Later on, we'll be joined by David Montgomery and Biklé, a husband and wife team who write about agriculture, soil health, food and microbes, bringing us very interesting stories and evidence on how we can grow a revolution by restoring life to the soil. Very interesting. Thank you so much all for being present and being here with us. And as you know, at Food for Sustainability, we work relentlessly to connect microbe science with real farming systems. For example, at our Soil Health and Function Laboratory, we analyze how soil communities are structured and how they function. We grow from bacteria to fungi, also envisaging enzymatic activity and nutrient cycling. This is where we test also bioinoculants, and we build with these tools strategies to rebuild a living soil, a living network that turn this very complex data into practical guidance for our farmers. Also, at Ampliaqua, which is our multi-trophic systems on the coast of Nazaré, we look at life in water, microalgae, fish and vegetables, sharing all the same loop. This gives us a unique place to study how aquatic microbes, including fungi, they help to close these nutrient cycles and stabilize production and the stress. And also at Idania Rural Food Park, we take all these insights and we put it into practice, scaling into the landscape. Soil and pasture diagnostics, they support regenerative practices in extensive and silvopastoral systems. And with this, we are able to link healthier soil biology to meet quality carbon storage and the vitality to the rural territories. And it's these three living laboratories that constitute the backbone of Food for Sustainability. And also, this is why we are here, and this is the context for our today's discussions. Just some house rules before we start. Please note that the sessions are recorded. So if you prefer not to appear, please feel free to keep your cameras off, but just a note to encourage you to keep with your cameras on whenever possible. This makes the experience much, much richer for everyone, particularly for our speakers. And again, a reminder that at the end of our webinar, you'll be able to receive your certificate after you complete a very short, simple quiz. Okay? I think we know the rules by now. So as usual, Sophia, I don't know if you're ready. Let's warm up. Let's fine tune into today's topic. And I would like to start by inviting you to come to Slido. Okay, so Sophia already has all the possibilities for you to join Slido. So you have the QR code, you can use it, or you can use the tab in your browser, www.slido.com, and in the hashtag box, just insert the code F4S Academy, and we are ready, once you have done that, to start with the questions today. And it's really good that we have this opportunity to warm up. It's quite cold from this end, so thank you. Sophia, Soraya, and Dimitra, which is the team behind that I never have time to properly introduce, but that prepare everything for you and make, you know, all these fun activities possible. So we already have, thank you, we already have our first question. Are you aware of the term Wood Wide Web? Do you know what this refers to? Is it the root system, the fungal network in soils? Is it about insect communication, or is it about bird communications? And come on, guys, we have more than 30 people in the room. Join Slido. Let's bring some emotion into Slido. Let's see if other people have different opportunities. This is very, very consensual, but the root system is taking pace. So a little bit of explanation. The Wood Wide Web is a popular science term that is used to describe the vast underground network, particularly in forest soils, that link the root of many plants, and often this can be different species, and allows the plants to share water, carbon, nitrogen, and very interesting, sometimes they even share defensive chemical signals. This hidden network plays a crucial role in forest ecology, resource distribution, and plant communication. At its core, it is formed by a mycelium of mycorrhizal fungi, whose fine ivy create these interconnections between plants. Very good. And you're absolutely right. Fungal networks in the soil, and they do this amazing job, you know, making plants even speaking to each other. Well done. Thank you all for taking part. We can move to the next question. So what do plant roots primarily exchange with their microbial allies? So what is the primary exchange that plants and microbes exchange? So starting voting. Okay, so this is very, very consensual again. So at the core of plant microbe mutualisms is a resource exchange that takes place in the rhizosphere, which is, as we know, the narrow zone that surrounds plant roots. Through photosynthesis, plants can produce carbon-rich compounds, mainly sugars such as glucose, and release a substantial fraction of these photosynthesis, sometimes as high as 30% into the soil and around the roots. So beneficial microbes, they become partners, and they include mycorrhizal fungi and nitrogen-fixing bacteria that use these exudate sugars as their main source of energy. In doing so, they help the plant access other scarce resources, with mycorrhizal fungi enhancing this uptake of water and nutrients like phosphorus, and the rhizobia converting atmospheric nitrogen into plant available force. So in essence, as you rightly point, plants are trading sugars for nutrients and water. So thank you so much. It's great to see that we have such a knowledgeable audience. It's raised the bar for our speakers. Last question. A healthy soil microbiome can improve human health by growing more nutritious food, avoid vaccination, transmitting probiotics, or curing some diseases. And it's true that we know that a diverse and active soil microbiome is essential for nutrient cycling and plant nutrient uptake. Plants, they grow in healthy and biological active soils. They tend to have higher densities of micronutrients. And we are talking about things like zinc and iron and selenium and some phytonutrients also, such as antioxidants and polyphenols. So what we are saying is that consuming food from such rich soils can indeed contribute to improve human nutrition and health also, meaning that a healthy soil microbiome ultimately supports healthier people by enabling the production of more nutritious foods. So it's growing more nutritious foods. And here at Food for Sustainability, we developed several studies that show that more healthy biological active soils are indeed producing more nutrient dense food. So we hope that we will feel warmer now. So it's time for us to pass the floor to our dear speakers. So I can see Paula, you're already here. Very nice to see you, and thank you for availability. So whenever you're ready, the screen is yours.
Thank you for your presentation and for your words, Claudia, and thanks for all the organization to invite me to be here and to share some of the work we've been doing for the past decades on this inoculation and soil health and microbiology. It's really a pleasure to share our small contribution to the field. I work as Claudia said, at Escola Superior de Biotecnologia, CBQF, in Porto. But we work in the field in many places because Porto is not perfect for doing experiments in the field. So what we do is very much around soil. Oh, just a picture here where we stand. We're here in this building, the glassy building that you see in the picture in the Porto campus. And I lead a group on environmental biotechnology and resources. And most of the things we do is around soil. We do soil with trees, forest, agriculture, production of bioinocular for animal plants to combat the abiotic or biotic stress. There is a lot around soil, but even what we do, if we look at the picture with water, and you've got there wastewater valorization, we also link to the soil because then we recover things from the wastewater treatment to put into soil, and it's also a lot of biomass going in. So, as was already introduced, the major point here is that soil is alive. So, in fact, it's what sustains a healthy soil is the microbiota and the macro fauna that it's colonized the soil. And if we do not have this in attention, then soil cannot support the services or the ecosystem services that it should provide us. And it's the only way to have a soil that is healthy, that can sustain all of the ecosystem services. And in the end, it's what affects what we eat, as what's also so nicely introduced. So if we want to have healthy food, we need to have healthy soil. If we do not have soil, we do not have food. So there's nothing to talk about. So everything of the ecosystem depends on soil. Is a bit much like what we see as we humans. What we have is also a range of microbiota. We have more bacteria than cells in our body, and it is the whole thing that makes us function. It's not only the visible organs, but also the invisible ones. And it is the same in soil. As you see, this actually represents the wood wide web that you just answered about, the connection between mycelia and root tree. So in fact, this is an example where you see that fungal mycelia goes deeper into the soil to places where the roots would not go, and there would not be so much efficient uptake of nutrients to the plants or to a tree, for example. So in fact, this is combating the soil loss that we're observing. It's estimates, you can read different numbers, but there is a number that in Europe, most half of the soil is there is loss. It's degraded. So it's even if it's productive, it's not healthy, and this leads us to think that we should do something about it. So in fact, in the past decades, we've been hunting for microbes from different soils, mainly from stressed soils and plants, endophytes. We've been hunting for bacteria. We've been hunting for fungi. We use mostly a combination of bacteria and fungi in our bioinoculants. And
Is it me or have we lost
No, I
Paula?
think we lost Paula, yeah.
Okay. Let's just give Paula a little bit to see if she can sort the problems from her end. Everything was working fine until now. Um, if not, Anita, would you be able to?
Yes, maybe let's give another minute for her to sort it out.
Okay, now let's see if these things happen. As you know, also the connections here in Portugal are still being restored after the dreadful events of Tuesday nights. And so that might give us also a little bit of some technical glitch. So please bear with us. I will just take this opportunity to remind you, Paul is here, to remind you that you can have the questions here in the chat. That we encourage you to stay until the end and to
I'm so
fill
sorry.
in.
Can you?
No worries,
I'm,
Paula.
I'm so sorry.
No,
The
no,
the
these things
hotel
happen.
internet went down, so I'm with my mobile now, so it should be
Okay.
okay. I'm really sorry. I'll just continue where I was.
Okay.
So just sharing again.
Yes,
Is it okay?
yes, first slide.
Okay.
Yeah, it's moving. It's perfect from our end, as if nothing had happened.
Yes, yes. I'll just
Okay,
forget about it.
great.
So please bear with me. So we've been hunting for these microbes, as I was saying, and one of the important things is not only to get them in the lab and do pot experiments, is taking it to the field. I think it's one of the most challenging issues about bioinoculants is how are they going to behave in the field. And we're hunting bacteria, especially those which have plant growth promoting traits. It goes for a range from like phosphorus solubilization to biocide production, and they help the plants to survive in stressful conditions. And fungi, like it was mentioned in the beginning, it's a symbiosis with plants, and we mostly work with ectomycorrhiza fungi, mostly mushrooms. So that's those are the, at least for isolation, that's what mostly we work with, and this is mostly for trees. If you're talking about crops, like annual crops, then we're talking about endomycorrhiza, which we also use, but mostly we target the ecto, which is also very crucial for soil health. So we try to combine these two and to design these bioinoculants, but it's not just a question of finding the right microbes. It's a question of being able to deliver them in the field. Is a question of being able to provide them to the farmers or to the forestry in a product that can be used, that has a certain shelf life. So all this is a challenge. We need to provide the product in the end. If you're targeting a practical application that is in fact usable and is compatible with the practices that a farmer or a forester has. So I will go through some examples that tackle some of these issues. And one thing that we do not do, at least in the field, is to use bioinoculants on their own. We usually work in big teams with people from agronomy and chemists, others, and we use different practices, bioaugmentation or bioinoculation, whatever you want to call it. Personally, I don't believe too much on that this works on its own. It works with crop rotation. It works with adding amendments. It works. You have to use a combination. There's no solution like one fits it all. So it should be integrated with a range of good practices, like Claudia showed some at the beginning. So it's not only about the microbes. So I'll just go now to different examples. And I will start with an example of soil degradation, fire, and ectomycorrhiza with bacteria. We've been working for some years with this type of stress, burned soil. With the inocula that we have isolated with microbes. So we went to forest, took some mushrooms, isolated the ecto from the places we wanted, and then we produced in mass. It's quite good for the ecto to be able to be produced without the plant. If it's the endomycorrhiza, you don't need the plant, so it adds an extra complexity. But of course, you need them for annual crops, for example. You don't. It's not going to be the ecto. And just to give you an example, this is, if you look at this, just the images, what happens in the nursery, because we're talking about forest plants. So you grow them in the nursery at the beginning, and then you transplant to the soil. And you see here, these are pine trees. And in this in this picture, you see not only a bigger growth when you use the inocular, but especially look at what's invisible, which is the root system that is better constructed. And it's not just about the stress that the plants are facing. It's also because you can have this biofertilizer as a contribution to have or to need less intensive practices of chemical fertilization of or agrochemicals, because the plant goes to the soil in the transplantation protected with the right biome. That's what we target. So it's easier to do it at the nursery. Just an example of pine trees. I have to say this was an experiment that I do not forget. We planted 10,000 trees, but this was for fire. It was in Serra da Cabreira. But a few years later, 2 years later, there was a fire, and our field was also devastated by fire. But we took a lot of good practice from what we have done, and this is just within the same experiment, the same inocula. You see here, they look so much bigger. Well, this is not real. I mean, the images are real, but we did an experiment where we used burnt soil as the substrate in the nursery. You don't do that really, but it's just to say that within a natural ecosystem, if you do this, the regeneration, if you have a healthy soil with the right microbiome, then you'll have, of course, a better environment for the trees to grow. The previous picture were with substrate. And this is another example, Quercus suber. We worked a bit with the Quercus, with another type of inocula, because these inocula are very specific. And you see these plants after some months, the control and the bioinoculated plants have a difference. But this is the real thing, is what happens in the soil. And if you look at the inoculated plant and the control plant, this is something different. And that's what you want. You want trees to grow to the field healthy, so they can stand up the transplantation. They can survive transplantation and establish themselves. And this is it's just, I'll only have a couple of graphs. We followed these plantations. This was planted in the south, very harsh conditions. We followed throughout the years, for some years. And what was, if you look just at the green, is the control. Inoculated plants are the blue ones, at least with our inocula. The yellow one, the bar, is an inocula that is a commercial one. So it was not, let's say, endogenous from the places. And what you see is that you continue to see the difference in height, but mostly you see the difference in diameter. For Quakers, you want to extract the cork, so you want to have a good and nice diameter for the trunk, so you have more and more cork. So depending on the plant, then you see what is your target and what you really want. But this is a major one. When we did these experiments we did with mycelia, it's a big issue, because you need really huge amounts of plants to be inoculated. We worked with a nursery plant in the south, trying to do it not with mycelia, with spores, and you get better results. But there is a lot of things to do in this field, because even quality control for inocula that you buy commercially is an issue, because you never know whether it's viable or not. I find it's one of the major issues in production. So now just a few examples for annual crop. This is a completely different thing. You're not going to a contained space like a nursery, inoculate and wait for your results and then put the plants in the field. No, you have to do inoculation in the field. Of course, you can have seed inocula. We don't do it. We do it in the field. And here you see an experiment where we were irrigating with the saline water, or with high concentration of salt. This one is in the lab, and what you see as you go from one set of bar to the other is an increase in the salt. So you have lower growth. The first column in each set is always the control, and then is the inoculation. And when you have the inoculation, either for the shoot or the root, you have much higher biomass in both. So these were, of course, these were inoculants that were selected for this trait, to protect the plants against the drought. You see here some pictures. This was done with the maize, but also what something we look at. I don't bring any example for the diversity of microbes, but you can look at enzymes, and you see here again, the first bar is always the control, and then when you have the inoculation and you check for soil health measured by specific enzymes, you get a good improvement on the activity. So in fact, the enzymes reflect the microbiota that you have in soil. So even inocula that is not, I really like the idea of the soil transplants. We're working on that. But even an inocular that is just like what you have in your yogurt, it's just one or two types, does affect what's happening in that rhizosphere and in the plant. And if I may show you another example of this growth promoting bacteria, this is something we do. And if we go back to the wastewater treatment, the leftovers, let's say, of a wastewater treatment plant is biomass, and we're working with a particular system where you have what is called granular biomass, which 80% of the biomass is EPS. So we recovered EPS, hexapolysaccharides, that have shown to have growth promoting traits. They're also good at absorbing water. And what we've done is to see whether the EPS or biochar, that is also much used in soil, as I say, we're integrated practices, and the inoculation of it, how would it affect the plants? If you see here, if you just look in this one, in this picture on the bottom, I don't know if you see my hand, you have a control here, and then here is the growth, this is maize, with the EPS, with these hexapolysaccharides that you extract from the water, the wastewater, the leftovers, let's say, the biomass, and this is biochar. Okay, we used to, I think it was 2.5%. Biochar, you cannot use much because it does affect it negatively if you want to do. Some people want to use biochar a lot of biochar but it doesn't work because it has the phytotoxic effects. And this is not shown by us, it's shown by many studies. But so here in these bars, the first one is always a control. Second and third is the EPS, and then the rest is the biochar, fourth and fifth. And what we've seen is that EPS is good, but if you further inoculate, is even better. So actually, we're using EPS as a delivering agent, and we would like to use it to increase shelf life and to have a product that is more viable in the end. So we try to link everything and not just the plants. But just to finish off, I would like to give an example of Recrop. It's a project that involved 7 partners or 7 countries, more than 7 partners, 3 from Africa and 4 from Europe. And we were targeting vineyards, maize, and medicinal plants, not all doing the same work. And this was mostly field work. So the 3-year project, it's also a problem for projects. 3 years is not much when you're doing agriculture or forestry, whatever. We did get an extension because we want to see this for at least 2 seasons in the field. And I'll just go through 3 examples. This is Galicia, Spain. This is an example for a colleagues from the group. They used our bacteria, one of our bacteria, and you see here the effect on the growth and on the mineralization of the nitrogen in the soil, which is related to soil quality, to microbial activity. In yellow is what happens for the enzyme in inoculated plots. And you can see that there is an increase in the quality of the soil health, let's say, when you have inoculated plants. And we use the same inoculant that we use in Portugal. In fact, exactly the same. These are vineyards that we inoculate in Portugal, and these are results from that we followed over the years. We looked at what I bring here is must production, is the must analysis, because these plants you're talking, it affects soil health. Okay, we're talking about wine, it's not food as bread that we need, but the inoculation affects the quality of the must. So all the phenolic compounds, acetic acid is affected by what you put as inoculant. And we're talking about bacteria and a mixture with endomycorrhizal ones that I haven't presented anything. And it's just another example of medicine and plants from our colleagues in Morocco. All this is corresponds to two crop seasons, and this was done with medicinal plants.
I think we're having connection problems again. What a shame. This was being very interesting. But it can give you some time if you have some questions you'd like me to pose to Professor Paula. Please have them here in the chat. Okay, this is how we we prefer to point our questions. Let me remind you that if you are interested about these topics, and if you want to know more, for example, we're going to run one edition on soil microbiome analysis. So it's to teach you how to interpret the results that come from genetic sequencing, the reports that are coming from the lab. We're going to have our next edition in March. If you want to know a little bit more about this course, let us know. Reach us out through our emails. We'll be very pleased to have you on board, and also to answer any questions that you might have. And also at Food for Sustainability, we have ongoing courses. So if you don't know what to offer as a present to someone that you care a lot, you can always offer this person the chance to know a little bit more about soils, what they entail, what they do for us, and what they do for agriculture. So we do have available courses, MOOCs that are constantly running. Also, just one final word for you. If you are attentive to our webinars throughout the entire cycles that we are going to run, we also have prizes for you. We're going to see who is the most frequent participant, and we're going to reward you for your presence and for your commitment with us here. Yes, the webinars are recorded. They are on our website. You can reach them through our website. We'll make them available in a question of days. You'll also have more about the speakers and about the work that they develop. Very conscious of our time. I think I'm going to press ahead, Anita. If you are ready, I will kindly ask you to come to the fore, to the screen, okay? And then Paula will join for hopefully later on, and we'll continue the dialogue with Paula. Anita, all good from your end?
Yes, all good. Can you see my
Okay.
screen?
Yes, perfect. Thank you.
I would like to thank you, everyone, and Claudia and all your colleagues for organizing this webinar. I think it's very interesting also what Professor Paula just told me, and my work really link into this. But I will show you a very different perspective, because I don't study single fungi. I actually study fungal processes, and I use aquatic system as my natural laboratory. So today I would like to talk about fungal decomposition and how to improve soil health by adding the decomposition component in it. So I work in a Limnological institute in Hungary near the Balaton. I haven't got any pictures like Paula did from our institute. I actually just did the presentation.
That's absolutely fine, Anita. You
Yeah.
can.
So today, agriculture faces a number of problems, but how the soil is degraded, it's one of the key problems, and all our food depend on it. A number of diseases is increased, which made our chemical dependence increase. The soil microbiome has been simplified by microbial communities, and when you have a large stress, droughts, climate change, or any pressure disturbance, we really face a problem as to how to feed ourselves. So the question is, how do we rebuild a resilient food system that will give us a much better food in the future? So, as Paula also said, soil health begins with the mycorrhizal network, and fungi in there do many work. So the fungi itself is not the fungi, the cup mushroom that we all think, because if we just look at the mushroom itself, then we only see the trees. The magic works in the soil, and that's when we can see the fruit tree. So fungi in the soil recycle nutrient, and as Claudia also highlighted, it drives plant communication, it drives carbon turnover, dictates carbon storage. It gives the plant the nutrients it needs, and it regenerates soil structure. Fungi, if you have abundant of fungi, it compete with pathogens, therefore it protect the plants from stress. It regulate the nutrient cycling through the decomposition process, and therefore fungi is much more than just a disease, and we really need to change that perspective that we worry about fungi. So without fungi and the fungal decomposition, the soils lose everything. Fungi structure the bacterial community, and I have had experience where I added a fungicide into a decomposition processes. It not only killed the fungi itself, but actually it made the bacterial community also pathogenic. So where there is forest, water, or agriculture, these are all one living system, and we need to treat it as that. Decomposition in water, agriculture, or in forest, this is what drives the nutrient and how much nutrient is available to all the plant. And fungi, it's inside the plants, inside the soil, inside water, and inside the air. It's actually inside the air that we breathe right now. They perform very comparable ecological roles in all these environments. Lakes and aquatic system are less intensively managed than agricultural system, and they also receive a continuous organic matter input. So we can have a clear link between decomposition, the fungi, and the nutrient cycling itself. In aquatic system, we can see how decomposition fungal networks function and organize it naturally. So, and if decomposition can maintain forest, lakes, it can also sustain future agriculture. So fungal communities operate as a network. And in lakes, you have fungi in plants, sediment, and water. And basically, the fungi reshapes how the decomposition pathway occurs. The more complex plant you have a greater diversity of fungi. And if you have an invasive plant versus just native plant, this all changes the fungal community structure and how the nutrients is being processed. So in my research that I'm talking today primarily about, I cultivated fungi from 5 different aquatic plants. I looked at the different structure. I looked at invasive versus native. And I actually had about 127 isolates, which equated about 60 species of fungi. across 3 phyla, 6 classes, 15 orders, and 27 families. But the fungal classification is still not very good. There are 40 fungi that we could only identify at genus level, and these are the fungi that really matter. These are the Talaromyces and the Cladosporium and the Trichoderma, all the fungi that is abundant everywhere. So what I found is that the greater structure an aquatic plant has, it has more fungi. And the local plant had the largest diversity of fungi. And that plant actually were able to grow over 150,000 cubic square kilometer. So each plant actually had a unique decomposer and then endophytic fungi, which they shared in the plants nearby. The second things I found, it was that invasive plants, how they restructure the fungal community. So near an invasive plants, I could only find Trichoderma species. And I don't know if you know Trichoderma much, but Trichoderma is a mycoparasite that actually eats all the other fungi, which then reinforces and enables the invasive plants to actually grow much faster. So Trichoderma is a strong competitor to all the other fungi. And the invasive plant actually reduced the diversity, which favored the invasive plant success. But if we have Trichoderma in an environment is good, but is also a sign of disturbance. So when the decomposition in a soil or water is disrupted, this fungal pyramid, it actually changes. When we use fungicides and we kill the decomposing fungi, then actually you have an increased number of pathogens. And actually this food pyramid tips upside down. It changes the function, how the fungi working. It weakens the competition, it changes the nutrient cycling, and increase how many pathogens there are. So it actually means more disease and more chemical dependence. So fungicide, in effect, it actually further disrupt the fungal balance, and the stressful environment favor all these pathogens. So managing soil to support fungal decomposition is what we can learn from aquatic ecology, is that the diversity stabilizes the decomposition processes and the nutrient cycling. Simplification increase the disease risk and the chemical dependence. Agriculture disrupt this network as it stands today. And we need to change basically our perspective to diverse plants and less disturbance, less tilting, adding food for the fungi, plant residues, manure, compost to promote the diversity and to avoid the fungicide, or just the timing of the fungicide should be really considered how it's used. And sustainable food system begins with fungal-driven decomposition. And I would like to thank you.
Thank you so much, Anita. Very, very interesting. And I also see that Paula has managed to join, so we'll also take an opportunity to have the presence of Paula. Anita, a very interesting presentation, and I will just ask one question, and because I feel that you have here an important message, that and we hear some echoes and some skepticism about this topic. So you have shown that fungal decomposers are actually essential for breaking down organic matter and building soil structure and fertility. But from a farmer's perspective, this is quite often a question of concern around fungi, mainly because they can be plant pathogens. So can you explain how this decomposer and also mycorrhizal fungi can actually reduce disease pressure and outcompete these phytopathogen fungi, and eventually what kind of management practice will help to balance this in favor of the good fungi in real farm soils?
I believe that you need to make you need to give a job for the fungi to make it busy. Fungi basically turns to pathogens when you have stress, when an environment is under stress, when conditions are not enough. Actually, a good fungi can turn pathogenic as well. So if we less disturbance and more work for the fungi, will make the fungi not pathogens at all.
And
So
so it,
you adding plant residues or plant nearby plants that will supply a good mycorrhizal network. Local plant with an abundant fungi would make that plant nearby more successful.
Yeah, and this goes back to what Paula was also presenting,
Exactly.
that you need this
Yes.
mixture of interventions to make sure that the system is still balanced. Is there any particular management practice you would recommend to farmers in a specific context to make this balance right?
I guess you need to look at each area individually.
Yeah, that's the challenge with farming, isn't it?
Yes,
It's
because
difficult.
you have the cork trees in Portugal and you have the olive trees that would be very different to a cornfield. But when you look at it, you have a tree, and when you only take the fruit itself, the tree will survive. And maybe every other year or every third year that will not produce that much food because it will put it down the soil, but it will later then just give it back again. So we really need to just find this balance and support the decomposition processes because as much as decomposition is not a beautiful thing, life starts when decomposition is completed.
Yes, that's an interesting also takeaway. It's not a nice thing, but it's probably the start of life also, and we need to respect that and pay a lot of attention to it. Paula, I don't know if you can hear us, if you can. Uh, Also,
Yeah.
Paula, thank you for introducing the topic of our next webinar, which is biochar. But can you join us? Cannot see you, but I believe you're
No, you
there.
can
Just
see I'm here somewhere.
okay, you're here somewhere. Okay, Paula, just one short question for you. While you were presenting, you mentioned that the challenge was to understand how bio inoculants behave in the field. So the lab is one thing, the field is another thing, and we hear a lot of concern by farmers about the
Mhm.
risks and consistency when using bio inoculants. So do you have an advice how to monitor, or which indicators the farmers should be paying more attention to these living products to see if they are actually working?
Yeah, that's a harsh question because I think one of the things that are missing in this in this field of for going to the market with these products is actually demonstration. It's I think there is a scarcity of demonstration, and as I was just listening, each situation is one situation, so it's very difficult to make sure that there will be reproducibility as well. But with living things, it doesn't matter whether it's bioinoculants or just crop rotation. It's very difficult to make sure that everything will happen exactly the same every year. So I think we just have to go with nature and see how it goes. But you were talking about um, indicators is actually one of the main aims of this project, and another one that we're starting in also in agroecology. It's what are the indicators that a farmer can use to measure, for example, the soil health. It's not easy. Many groups are developing that. So for now, it's mostly to take a bit of soil and see whether it looks good and it has the worms and the living things that should be there. Okay, I don't want to make this too simple, but it's a good thing. There are things that are very simple and that are an indication of a good soil.
Of life in the
Okay, I
soil
don't
and
even know if
function.
that was the question, but okay, I tried to. I'm very skeptical about saying, okay, you should add this or that or that, especially as to commercial or lab or coming from university. It doesn't matter because this problem of shelf life of these products is still an issue. You need to
Hmm.
make sure that they stay high quality and viable in the shelf for a long time. So when you had, for example, soil transplants, which is actually in the past a bit what was suppressive soils. It's something that is feasible, of course, not in the large scale, but we have to go with also with practices that we can introduce in a normal life of a farmer. And that's why we talk a lot with the agronomists, and all the projects are with the farmers and associations, because we're not agronomists, we're biotechnologists. So we're
Hmm.
just trying to fit in the puzzle. But we okay, this is also most of the times we're doing this because the soil is not healthy, or is not in is stressed, because if it wasn't, then the normal cycling will
Hmm.
occur.
Yeah, you mentioned something that's quite important, is that the soil is under stress. The question
Mhm.
is that probably more and more often our soils are under stress.
Yeah.
And, if it was at an equilibrium, eventually you don't notice,
Yes.
but under stress, you need this
Mm.
microbiology to be functioning and supporting and assisting it. Some people call it microbiology is like an insurance for when things go wrong. And
Yeah.
more often things are actually becoming more challenging. You also had the number that 45% of soils
Mhm.
um, are actually
Mhm.
degraded. Okay, and it's getting one final note, just because you didn't have time to finish your presentation just in 2 minutes, about soil transplants. I know you're passionate about it and you have done some
Yeah.
work on that, just to our audience.
Well, we really going on into that. It's not okay. It's just making the say the inoculum more complex, because it's not just one bug that does the job. But it is a very interesting and challenging field, because it's you can't just start with digging soil and putting it somewhere else, or there are questions to this. But uh, yes, that's one of the things that we should go into to sometimes probably this is more practical even for a farmer, for to be used. But we haven't worked so much on that, but I think it's the way forward to synchronize more the communities. Uh,
Hmm.
Even when I talk about trees and the crops, okay, they are very different things, but the ecosystem is made of all those trees, the crops, so is the community functioning. Um, but yes, I'm not saying too much, but soil transplant is I think is a way forward as well as
Okay.
many other
Don't worry,
things.
don't worry, Paolo. You're already pointing us in the right direction. Paula and Anita, the scientists, and working a lot towards new solutions that can actually assist farmers in the field. We know that this type of science is fairly new in terms of applications, and it's good that we are aware of some of the problems, and we need to have scientists working towards that solutions. It was very, very helpful. I can see that David's already looking at hearing us very, very attentive. David, good morning for you. I know it's and Anne, good morning. I know it's early morning for you, so I will invite you to join in and to tap into this conversation. We hear a bit of feedback. Do you have two pieces on? Maybe Anne?
No, so can you hear us?
Yes, yes. Oh,
Okay, but there's feedback.
no, it's fine. I will shut up. It's just so
Oh,
that
okay,
I
good.
can
Well,
only
good
hear
morning
you.
and greetings from Seattle. I hope you all are doing well. I'm Dave. Anne, my wife, is next to me, and we're going to share. I take you want us to go ahead and start. We'll share a bit about what we've been doing literally for the last 20 years in thinking about soils and healthy soils. I'm a geologist by training. Anne is a biologist. What do you get when you put geology and biology together? You get soil. So we've been working in this area for a while. I'm going to try and share my screen here, and what you're seeing is that we don't tend to use Microsoft Teams.
Okay.
But anyway, basically, Anne and I have worked for literally almost the last 20 years on this series of books that charts our development and understanding really of the role of soil degradation in the past. When we wrote Dirt, the Erosion of Civilizations, that started us off on it. It's a book that is focused on how societies have degraded their land that in the past, defining the problems. The Hidden Half of Nature talks about the role of soil microbiology in soil fertility, a lot like the previous two speakers have been speaking about. Growing a Revolution looks at how regenerative farming can turn the degradation around. And What Your Food Ate, our most recent book, looks at what this all means for what gets into our food and human health. So we're going to give you sort of a quick tour through these books. Happy to engage in questions after. But the good news is we wrote most of what we know about this all down in the books. And when we summarize the Dirt book, it's a look backwards at the history of soil erosion. And the take-home messages are really that societies that don't take care of their soil don't last. That's a theme that has lasted literally through multiple cycles in different parts of the world, in many regions of the world. And the thesis of the book is that soil erosion and degradation, the degradation of soil health, soil organic matter, the life in the soil, has limited the lifespan of past societies. Not that soil erosion wiped out societies, but it set the stage upon which it rendered them vulnerable and not resilient to the kinds of changes that do tend to end societies. And one of the problems that we identified in writing Dirt is that soil erosion is not just a problem of ancient history. I also compiled data from several hundred studies around the world to look at what rate are modern farms eroding at, how fast are we losing topsoil on their agricultural lands around the world. And what I came up is that in global average is that it takes modern agriculture about 20 years, a couple decades, to erode an inch of fertile soil. That's about a millimeter per year. And that's actually pretty slow in the pace of things that most people think about. But to a geologist, that's screamingly fast, because it takes nature 500 to 1,000 years to replace that inch of soil that we've been shedding in just a few decades. And that's a problem that is not just characterizing past societies. The UN's Global State of the Soil Assessment from back in 2015 concluded that we're losing about 1/3% of our global food production capacity each and every year to ongoing loss of topsoil, soil erosion, and the degradation of soil organic matter, and the degradation of soil health. And that 0.3% doesn't sound like a big number until you think about, well, let's project that out to the year 2100. It would project that if we're losing 0.3% per year, we'd be losing about 30% or 25% now that we're partway through the century of our ability to feed ourselves globally at a time when our population is projected to rise by anywhere from 1/4 to 50%. This is obviously a big problem that humanity faces. I'm delighted to see folks like the people attending here thinking about soil health and practices that could rebuild soil fertility as a consequence of farming. That's when, after I wrote the dirt book, one of the things I was very curious about was whether or not we could actually rebuild soil fertility on economically viable, highly productive farms. So I visited farms around the world that were practicing regenerative agriculture that could actually rebuild soil fertility remarkably fast. And what I came away from that experience with is seeing how farming and ranching practices could actually put a lot of carbon back into the ground. That's the good news. The bad news is we won't be doing that through our conventional farming and ranching practices because they degrade soils. We need to, in other words, reform agriculture to be a regenerative practice rather than a degenerative practice. And I know that that's possible because I've seen farms around the world, from small subsistence farms to huge, highly mechanized farms in the American Midwest, that have done it, have rebuilt the fertility of their land. And how did they do it? Well, I summarize that by ditch the plow, cover up, and grow diversity. And a little more formal way to look at that is, what were the commonalities between the practices of these regenerative farmers in lots of different contexts and settings around the world that had rebuilt the fertility of their land, of their soil, and the health of their soil as a consequence of highly productive, intensive farming. And to a person, these farmers were all had adopted practices based on the principles that are up on the screen now, of minimizing chemical and physical disturbance. So trying to minimize disturbance of the soil, which would help promote those fungal communities and bacterial communities that were beneficial. Always keep the soil covered with either a cover crop in between cash crops. In other words, don't leave bare plowed fields vulnerable to erosion by wind and rain. Diversifying crop rotations, or diversifying the amount of crops standing in a field at one time on some of the smaller subsistence farms around the world I visited, and then reintegrating animal husbandry into agronomic cropping practices where it can actually work. The individual practices farmers used, varied based on where they were in the world, their climate, the crops they were growing, the equipment they had access to, but the principles that they were operating under broadly were characterized by this set of regenerative principles that practices could map under. What could that do to the soils? Well, I could show you a lot of examples of soils around the world that farmers had rebuilt the fertility of. This shows you one from the state of Ohio in the United States, where the farmers started with soil like this khaki material on the right. Back in the 1970s, they basically went then to no-till farming, minimized disturbance, weaned themselves off of agrochemicals, started to grow plant cover crops, grow a diversity of crops, and over the course of literally about a decade or two, converted this khaki soil into this rich brown chocolate cake. And I've never met a farmer who would rather be farming this organic matter poor, devoid of life soil rather than this rich, healthy, fertile soil. And that's not a unique experience. Anne and I orchestrated a study of farms across the United States where we took 10 pairs of regenerative and neighboring conventional farms, and we had them, we analyzed their soil and their soil health. The regenerative farms had been farmed for anywhere for 5 to 10 years with practices that have followed all three of those principles: minimizing disturbance, having cover crops, keeping the ground covered, and growing a diversity of at least 4 or 5 crops in a crop rotation. And what we found is that consistently, the regenerative farms had on the order of about twice the organic matter as the conventional farms. They doubled their organic matter in less than a decade. And the soil health scores using the Haney soil health test had values about 3 times higher. So these regenerative practices were putting carbon back in the ground and building healthier, fertile soils. But that's not all they were doing. We found that across the board, these farmers that had adopted regenerative practices had higher profits than their conventional neighbors. Why? Because they were harvesting comparable yields and using less fertilizer, less pesticide, and less fossil fuel to actually grow those comparable yields. So they were spending less to grow more, and that's a recipe for a more profitable farm. That's what started turning me into an optimist on the issue of soil regeneration at a global scale, because if it works out for economically for farmers, then the environmental benefits can flow naturally from the behavior that may follow, and that increases soil carbon, water retention, and results in less offsite pollution from using less fertilizer and pesticides. And at this point, I'm going to turn the discussion over to Anne, because she's going to lead you through the second 2 books that she co-wrote.
All right. Okie dokie. So we're going to zip on through things. And I think you can sort of get the sense that through the course of research writing these 4 books that we were definitely on a journey to learn more and more about soil and how we can begin to work much, much better with it than we have. And I would say, you know, ironically, this started with really learning to see what is invisible. And that began for us actually with, well, what can we see before we see the invisible? And it was our dead dirt. This was dreadful. This was our first home in Seattle. And I was desperate for a garden. And look at this soil. Now, I will make a small confession here. This was a while back, but it's embarrassing. Neither David nor I thought in our own yard to take a closer look at the soil, except when we got to this and I said, oh no, this is never going to do. I panicked, frankly, but I also knew that organic matter might be a way to get us out of that trouble. You see, I had done a bit of gardening, and organic matter always seemed to help. I couldn't say exactly how, except that over short periods of time, even longer periods of time, the soil seemed to respond and the plants seemed to respond. So these are just some examples of organic matter that I collected nearby my wannabe garden. And interestingly, all this stuff, it was either free or it was cheap. And I really like this idea that we've got all this organic matter laying around, and it's on a lot of farms as well. And if we can better utilize it and be returning it to the soil, that will be a good thing. And as you've heard from many, many speakers, organic matter is absolutely fundamental to soil life. So here we are, about 10 years later, that horrible soil that sent me into a panic attack there on the left, and that lovely soil on the right. And all right, so what else was really going on? Well, this is what was happening above ground. And I will tell you this, there is not any fertilizer, there are no other synthetic inputs, there are no biocides. This is what the botanical world in a garden on a farm, this is what it can do. So I was adding a lot of organic matter. I was enlisting Dave when I could. I need help. We have this organic matter. We need to mix this up into mulches, get it out on to the top of the soil. And what I did not realize, and this takes us into the invisible, is that we had a lot of help from microbiomes. Now, there's a lot that we could say about microbiomes. I like these 3 key points, and I want you to remember them. This word indigenous, that means that these microbes are supposed to be there in their hosts, in and on their hosts. That might be the soil itself, the plant body, the human body, say cows, sheep, dogs, cats, all of it. These microbes are indigenous to their hosts. Now, we heard a little bit about inoculation, and here we're sort of blending the indigenous concept with, you know, help from us. The whole point here is that without microbes and without a microbiome, a host would be profoundly unhealthy. And part of that is because these microbes, these communities of fungi, bacteria, archaea, viruses, and more, they are making nutrients available for their host. And as we are learning, not only making them available, they are making nutrients in many cases as well. And this all has a profound effect on host cells and genes, which I'll get into a little bit toward the end of this talk. So I want to take you to the wild and alive place beneath our feet. This is the invisible world. And so all of these scientists that have been talking about their experiments and their research and all of these graphs about how fungal and bacterial communities change when we start feeding the soil with organic matter, keep this image in your mind about what is really happening there beneath our feet. So this is the world out at the tip of each and every root and root hair on a plant. And what makes this world work, in part, here we have our root tip, And we've got fetching fungi. These could be arbuscular, mycorrhizal fungi, the endophytic types, and so on. Here they are, one end in one way or another, attached to communicating with and feeding that root, the other end far off in the soil, collecting what plants need. That is why I call them fetching fungi. They are there and they are helping. And what feeds all of this are these plant exudates, right? So plants, of course, have monopoly on sunshine. They use that to produce food for all of these microbes in the rhizosphere. This is fats, this is carbohydrates, this is proteins. Folks, this is meals for microbes. That's what exudates are. And down here at the bottom, what we're seeing is that bacteria, for example, are consuming these exudates. They need to eat, they're an organism. And we often call their byproducts waste products. I want us to get that term out of our minds because these products are absolutely essential to crop health, crop defense. You heard about some of these things. It's plant growth promoting hormones. It's signaling compounds that can stimulate the plant immune system to deal with pathogens. And so what we have here really is a micro ecosystem out here at the tip of every root and root hair around a plant. And this has been going on a long time. It is said that the most life-dense place on our planet is the rhizosphere. This is this very narrow zone around the root system. And it's not just been going on for a couple of decades. We're talking about some of the, going way back in the early days of plant evolution, some 400 million years ago. This is ancient, this is time-tested, and this works. Why would we not use this in agriculture? That's my question. Okay, this is also, to sum it up, this is a health plan, folks, for soil and crops. And this led David and I to another question and to our the next book, which is What Your Food Ate. And here it is, right up here. What if the way we farmed nourished these microbiomes, the crop body, the soil body, and our livestock as well? So What Your Food Ate was really asking questions and making these connections. Does it matter how we farm? And I think folks here in this webinar, through all of the presentations, you know it matters a great deal how we farm. And I want to share with you sort of how this all wraps up and links up. How does soil health relate to crop health, livestock health, and human health? And there is a lot in What Your Food Ate. I want to focus on sort of something very fundamental that David and I came to with all of the research that we did to write this book. And it has to do with what we call the Fab Four. And so these are micronutrients. So this is things like iron and zinc, as well as vitamins. And I like the word micro up here because it reminds me of the microbes. We need them. These are small quantities of vitamins and minerals. Minerals in particular, say iron, zinc, selenium, and so on. We need them in very small amounts, but they do very big things in our bodies. And plants, of course, pull this up into their bodies with help through their microbiome. And so micronutrients are one thing. Fat balance, I won't be talking about animals for this presentation, but in particular, we're talking about the omega-6 and omega-3 fats. Phytochemicals, you know what these are. These are all, this is a vast array of compounds and molecules that the botanical world produces. And we're very interested in those, as I'll get into, because they relate to our health. And then once again, here we are, microbial metabolites. Now, another thing about the Fab Four that I want to emphasize here, they are only found in the plant and animal foods of the human diet. You saw that, right? We're not plants. We cannot, human biology cannot make phytochemicals. We are not the body of the earth. We cannot make iron. or zinc. We're certainly not a microbe. We cannot be making those metabolites. And even the two fats I mentioned, the omega-6 and omega-3 fats, they only come to us through primarily it's animal foods in the human diet, but also some plant foods. And I want to make this point as well. The Fab4 fats are a bit of an exception here. They don't so much as actually build our bodies, say, the way proteins, carbs do, but we need them for normal function. Our human biology uses the Fab4 in a variety of different ways. And I want to say here, make this point too. We often are faced with, you need to be super, you need to be superhuman, you need to be over the top, you need to be great. Actually, folks, what we need is normal. We don't need disturbed, we don't need abnormal, we don't need all of the perturbations that are out there. Just normal is just fine. We in our microbiome can do great on that. All right, I'm going to return to the study that Dave mentioned briefly, these 10 paired farms. And we sampled the crops there on the left, cabbage, corn, and so on. And we took a look at phytochemicals and micronutrients. And there at the top in the green, what we're finding, I want to know, these are average differences. So it wasn't that the regenerative farms always had higher levels of these phytochemicals, but on average they did. Wow, I like this, 15 to 20% higher. Why? It's helping our bodies function normally. These micronutrients, 15 to 30% higher. I like that as well. So this tells you what can be happening when we have a normally functioning soil, which is to say, all of the fungal communities, bacterial communities that the folks before us were talking about, these are where we want them to be in terms of their relationship and in terms of their role in the ecosystem with plants. And Dave already covered the soil health scores. So what does this look like on the ground? This is Singing Frogs Farm. This is out in California. This is our version of a Mediterranean climate. It was hot on the day we visited. We had temperatures in the 90 degrees Fahrenheit. And I was amazed at how well all of their crops were doing. And so I like seeing the soil covered. I like seeing the diversity of crops. And these are brassicas. These are not crops that, normally grow great in the summertime, but here at Singing Frogs, they were doing fine. And then I think it was the previous presenter who said, sometimes it may not be entirely scientific, but we know a good soil when we see one, right? Look at the level of organic matter and carbon in Singing Frogs Farm, that adjacent farm. I could not enough time to tell you a story about that, but the soil, look at that. That's not at all what we want. All right, the Fab4 in our bodies, what is going on? I'm going to take you through the last part of this, just with an example of phytochemicals and microbial metabolites. So those crops, we want them suffused with phytochemicals and the fiber. Phytochemicals in part are bound up with the fiber of whole plant foods. And it turns out that the human microbiome, all the communities of microbes in our bodies thrive on fiber and phytochemicals. And most of the human microbiome is, forget about the stomach, that's way too acidic. Most of our microbiome is down here in the lower reaches of our digestive tract. So what happens is we're consuming all these dietary phytochemicals, and I'll say this too, depending on the diet of animals, we can also take in phytochemicals through dairy products and through meat, provided those animals have been out on a pastured diet. So not inside of some kind of a facility eating the equivalent of ultra-processed foods for animals, out on pasture. All right, the human microbiome gets a hold of these phytochemicals and fiber, and you might think about this as our rhizosphere. And what goes on there in the rhizosphere, right? we've got lots and lots of interactions and exchanges, these microbial metabolites, which are not waste products. They are things that our body needs. So this is what the human microbiome can do with the human diet, make metabolites that have all of these effects over here on the right-hand side of the slide. There are tens of thousands of microbial metabolites. We do not know all of them by any means that the human microbiome makes that begin interacting with our own cells, our own tissues, that in the end can have these effects. If you're curious about this, we go into this a lot more in the hidden half of nature, but there is one fairly well-studied
And
group of
we are reaching the end of our webinar.
Okay, wrapping up here. This is really part of a person's health plan. And we've thrown a lot at you, and you could say it just like this: if it's good for the land, it's good for us too. And the story continues because new book coming out this coming August, Regen. Look for that. And we are done and happy to take questions. Thank you.
Thank you so much, Anna. Thank you for respecting the time of our webinar. So for the time being, if I can just ask you to close your mic. I hear a lot of echo. Thank you so much. Just because it's really, we are approaching the end, and before I turn to the questions, I'm going to share with you your opportunity to get a certificate, and I'll ask the team also to place in the chat the link. And just to say, and David, thank you so much for bringing such an important topic. And I think there's also the other side of, looking after our soils, is that we are actually looking after the human health. And we have done scientific studies about the nutrient density, namely what you mentioned about the omega-6, omega-3 ratios when the meat is coming from regenerative pastures. So this is really, really interesting. I was going to have here a question. I was quite curious about the regeneration that you did, that you showed in your garden. And actually, this relates to a question that is very important to farmers. How long did it take for you to regenerate that system? And what was your starting point?
Let's see. So I think you saw the picture of that soil, that dead dirt at the beginning. And
exactly.
Yeah. And within 2 or 3 growing seasons, as the mulch got laid out about 2 times a year on top of the soil. So this was not dug in. It was out on top of the soil. Plants were in the ground. Now here in the Pacific Northwest, we get a lot of rain. And so that sped up the process, that nutrient cycling that by which dead plants help feed more life. 2 to 3 growing seasons, we were beginning to see a micro thin layer of organic matter on top of the soil. And it all depends. I think one of the previous folks had said, every place is unique. So depending on your types of organic matter, how you are incorporating that, precipitation, temperature, and so on, that will determine how quickly you can turn things around. And so it can begin very quickly. Maybe your eye doesn't catch on that immediately, but it was fairly quick.
Okay,
I saw similar things with farms around the world where farmers would start to see observable changes beneficial to their soil within a year or two. And they would basically, their yields, they would have an initial yield dip, but their costs would go down even more than their yield went down. And then their yields would come back up within about 2 or 3 years as they started to rebuild soil health. And across the board, I saw that within about a decade, there were radical changes to soils, so that you could, those sort of the khaki to really rich black soil kind of changes. So, the couple years to a decade could be transformative. It's much faster potential than I was ever taught in graduate school. for rebuilding
Okay.
soils.
And it's a process that we need to go into, because the challenge for the agriculture in the future are huge.
Yeah.
And I also have a question. I'm so sorry, David. Can you just close your mic? It's very confusing for me. Thank you so much. And because agriculture, is facing a new challenge, I have a question for Paula and Anita. So from your scientific background, which one or two research question about soil fungi do you consider is the most urgent to be answered in the next years, and also to address some of the problems and the challenges that David and brought to us? So research questions, what we need to know about, in your case, soil fungi, that you think is more urgent to answer, so we can deal with some of the coming challenges, like, drought or too much water, or, degraded soils.
The soil fungi needs the organic matter that Anne was talking and David was talking about. As soon as you put the organic matter in, the fungi will come and it will do the job.
Okay, so you need, before everything else, you need to have
Yes, in
organic matter.
agriculture you can plant in any plant that sheds a lot of leaves. That would make it natural. You don't even have to do the work. If you just plant the shedding trees or yeah.
Okay, good. So plan for more organic matter. And
Yeah.
Paula, one final view from your end, research questions. I know you do a lot of science to assist farmers.
Yes, you were speaking about the organic matter. Actually, even in the project I presented, the first question was how to increase the organic matter. That's a problem worldwide. I didn't show that the numbers on that. It's not also easy to measure or to compare sometimes, but we're looking mostly at the soluble organic matter in soil. Through these kind of mixed practices. I really like the presentation also of the second one. I could not hear because
Anita.
I was unconnected, of the David and sorry,
David and then?
view. It was really interesting. It's the whole community working. But for fungi, we do know that all these mycelia and all the structure of the communities that are so important for erosion and for all. If you talk about mycorrhiza, are destructed by chemicals. That we do not need to prove that. We've done many experiments on that, but everyone knows if you put pesticides, it's so obvious. The community it comes down very, very, very fast. And even with other practices, with the digging soil and everything, it destroys. And you do not need to go to the forest to see the mycelia, because it extends through the soil. So it's a combination. And you also need the decomposing fungi. It's the natural, it's the circular economy of working. So we cannot really look into just one component. But if you ask me what in the last two projects we're keeping with working with the Mediterranean countries, with Africa, with in another project of agroecology, and organic matter is the key question. Then everything comes down from there. And you need a good structure. I really like the pictures, the images of the soil that that the last presenters shown. So when you say how can a farmer look at it, it's just sometimes you just need to take a bit and just look whether it looks nice, smells good. And when I talk about biotechnology, and I'm really a fan of that, and really exciting for me, it's a push, because we just restoring what should be there. If it was not there, we do not use any microbes that come out of place. We go to nature to get them. So in the first place, they should be there. Biotechnology is a push. And I hope the conversation in a few decades from now is very different because we're
Okay.
giving it a push. It's like when there is a person that is ill and needs medicine. Okay, this is natural medicine. So I'm just saying this is not what we think that we should be always doing. Is a push to go further with other techniques, but we need to restore soil to the a good condition, and so we don't need so much help from humans, let's say. I mean, in a way that we're devising these solutions, but it's just restoring what is there. It's nothing else. It's just
Because
putting it
we've done
back
so much
because
harm, isn't it? So now we need
yes,
to intervene again to
it's
put
the
the system
same. It's like
back
sometimes
to
people think that is
functioning.
magic. It's not. Even when you
Mhm.
treat water, wastewater, that's something that I really like. We're not putting anything special in the pot. We leave too many people in the same place. Nature cannot work at the pace that it can provide us all with everything. So, okay, we're not talking about that, overpopulation in some place, nothing like that, but we're just giving nature a helping hand because it's nature that does its job. And microbes are the foundation. My favorite class that I teach is environmental microbiology. Students do not like it so much, but for me, it's fun because it's circular economy as at its best.
Okay,
So,
and it's fun for us all. I think I'm going to take that point, and I need to close this webinar
sure. Sorry,
now.
Claudia,
So
I get
no worries, no
excited with
worries. It,
this.
it was fantastic. I will take David's an expression that let's start here, a growing revolution. Okay, at
Yeah.
least as a community, let's do, which each we need its own department, its own angle to contribute, that we spread this word, that we have more and more people engaging in this movement, understanding and respecting soil. That's, everyone is very aware of water and very concerned about the air quality, but no one thinks about the soil. We are all very concerned about our health, but we don't think enough about the food that we eat. So let's keep coming. Let's push this message. For me, it's really a thank you for your presence. It was really, really a pleasure. We will be back in 2 weeks' time with a new series. We'll be looking at biochar. Let's learn more about biochar. And Paula, thanks for, unveiling a little bit that biochar is not the wonderful and savior of everything. So we will look into it starting on February the 11th, I guess, right, Sophia? So I hope that you're joining us,
Well,
and February the 12th. Thank you so much that you joined. For my end, I will say bye-bye for now. It was a pleasure to be here. Thank you so much to our speakers, those that wake up very early, those that had internet problems, and also to you, Anita, for a brilliant presentation, and to our participants, of course. For me, it's always a pleasure to have you here, and we are very, very happy with your commitment and your engagement with us. So I'll see you in 2 weeks' time. Bye bye.